Hi, my name is Katie Lankowicz. I'm pursuing a master's degree in fisheries science here at the Chesapeake Biological Lab, and my advisor is Dr. Hongsheng Bi. I do research locally here in the Chesapeake Bay and in the Bering Sea in Alaska, but it all focuses on spatial ecology. So that's the study of where organisms are, when, and why. My local research in the Chesapeake Bay focuses on where forage fish are in the Patuxent River region. And it involves taking a small research vessel and attaching what's called an eris to the side. So essentially, this is a sonar camera that I'm putting into the water, and like a flashlight through a dark room, it's able to pick up anything that passes underneath its beam. From there, I take all these images and then count all the fish that I see pass through my field of view. I'm able to get an exact geolocation and some environmental qualities like water temperature and salinity for every fish that I see. My results are preliminary, but we found that they do prefer to be in creeks as compared to the deeper river channel. When I do my Arctic research, I have my eris attached to a tow sled that also includes something called the zooviz. This is a zooplankton visualization system. It's essentially an underwater microscope, and it picks up many images a second of all zooplankton that pass through its small field of view. When I combine these two data sets, I get both the large macroplankton, like jellyfish and sometimes even fish from the eris, and the smaller things that they might be eating, like phytoplankton and copepods in the zooviz. At the end, I have so much data, about 22 terabytes. All of this data has to be processed, and I'm still doing that right now. I will be cross-examining where jellyfish are, where Alaskan or walleye pollock larvae are, and where everything that both of these species are competing to eat are. Eventually, for my thesis, I'll be tying in a little bit of particle image velocimetry. I have 3 high-speed cameras set up around a tank of water and a laser light sheet passing through the tank of water. Now what I want is for small larval fish to pass through the laser light sheet and be caught on camera. So I'll be getting high-speed, high-frequency, high-resolution photos of these fish's bodies and the water movements that result from their motion in swimming. Many people don't like jellyfish, but we should also care about their environmental impacts in the Bering Sea. Jellyfish clog fishermen's nets and actually also can eat Alaskan pollock larvae, and Alaskan pollock is the largest fishery by volume in the United States. Whenever you stop at a fast food restaurant and get a fish sandwich, chances are that's Alaskan pollock. So, in the same way that we need to budget for a healthy population of forage fish in the Patuxent River, we need to know how many jellyfish there are in the Bering Sea so we can maybe one day be able to budget their abundance and distribution for a healthy environment.